So, hello everyone. This is the fourth occasion now of CarboHyde's Masterclass, and I'm really happy to have you all here today. As usual, we'll talk about our topic today, which will be using cyclodextrins in biologics formulations. This will be about 30 to 40 minutes while we go through the slides. And after that, you can raise your questions, or even during the talk, you can raise your questions in the chat box that I'll try to answer, ideally related to topic, but of course, as time allows, we can also discuss anything else. So, okay. So today's agenda. Uh, what kind of biologic formulations we'll talk about with cyclodextrins? After just very a basic overview of what cyclodextrins are and how they are used, we'll see how they are used in proteins, how they are used in vaccines, other biotech uses, and how they are used in gene delivery. I'll try to show you as many commercial examples as they are available. Although we must say that the use of cyclodextrins in biologic products is still considered an emerging field compared to the small molecule formulations, but as you'll see, it contains a lot of opportunities and possibilities and a lot of stuff to do research about. So, cyclodextrins, if someone doesn't know them, these are ring-shaped sugar molecules, oligosaccharides, with a structure of a truncated cone or a donut, if you like it better. And the peculiarity is this part, the cavity of the cyclodextrin, which can encapsulate different types of products. And the marvel of this encapsulation is that as long as the complex exists, technically the original properties of the guest molecule are masked, and this results in a lot of advantageous features like solubility improvement, stability, movement, taste masking, odor masking, whatever. So technically, as if your guest molecule wouldn't even exist, the cyclodextrin, like a blanket, covers it, it disappears, and when the cyclodextrin releases it, of course, because it will release it, it will get back all the original properties unchanged. Traditionally, cyclodextrins are used by this many industries, as you can see, and many more, but of course, including pharma. And the typical mainstream application is for small molecule formulations, but nowadays we see these new novel applications. Some of those are on the table today. So using them as active ingredients to formulate different biological products, to develop targeted therapies or personalized therapies based on cyclodextrins, use them in diagnostic fields or in different tools in the biotechnology. So, how the cyclodextrin works, everything goes back or goes down to this inclusion complex formation, which does sound very simple. Cyclodextrin A cyclodextrin cavity can capture a typical small molecule that's usually roughly the size of the cyclodextrin that it can encapsulate. This is a non-covalent interaction, as you can see here. This is an equilibrium, so the cyclodextrin releases very quickly and efficiently the encapsulated small molecule. So there is always a continuous association and dissociation happening in the system. But how does this relate to so it's of course easy to understand that this is happening with small molecules, but what is happening with large molecules and how does this differ? Because as we'll see, it differs from the small molecules. So let's start with proteins because maybe that's the easiest to understand. What's the problem with the proteins? Of course, the problems are not the same as with small molecules because proteins can aggregate. Proteins are usually administered in highly concentration, or at least ideally administered in highly concentrated solutions, which have a high viscosity and a poor injectability. Proteins can absorb to the container surface or the container wall, and some proteins, not all of them, but some, have limited water solubility and can degrade. So they're rather sensitive molecules, we could say, and not easy to deliver and formulate. And ideally, of course, a magic excipient like a cyclodextrin, okay, it's not a magic excipient here, but pretty useful, can prevent all of this happening. So let's see how. Uh, Here we see a native protein, and usually upon some stress, there is a misfolding of the protein, which is followed by aggregation, degradation, or in some cases, oxidation, depending on the nature of the stress. And if we combine the protein with the ideal cyclodextrin, and of course, in an optimized formulation, the cyclodextrins can prevent all of them. Um, but how is this happening? So there are two types of interactions happening here, and one is the same as it typically happens with small molecules. So we can also talk about some molecular encapsulation and interaction with the cyclodextrin cavity, typically with the hydrophobic amino acids that are on the surface of the protein. But on top of that, we can also see that there is a kind of surface effect, a surface covering effect, like those of the polymers. So uh, cyclodextrins have these double mechanism of action when it comes to interactions with proteins. Of course, unlike with small molecules, we're not talking about one-to-one or one-to-two interactions. Here, the proteins are huge, so many cyclodextrins are interacting with the protein at the same time. But we know from evidence that this is beneficial to formulate the proteins. As you can see, this is fairly new, but not very new findings. So formulating proteins with the cyclodextrins. Most of the first papers were published in the early 2010s by Novartis, and then later these were followed by a lot of patents. What I also have to say is that despite the many and tremendous amount of literature on the topic, still there are no cyclodextrin-based protein formulation, pure protein formulation like a monoclonal antibody formulation on the market. Uh, but there have been a lot of attempts, and as I said, there are a lot of patents, and there have also been a lot of clinical trials to formulate proteins and bigger peptides like insulin to deliver them, even via novel administration routes, like can we deliver intranasally a protein or orally a protein when it's encapsulated with a cyclodextrin. And so is a nice example that it worked. And this development actually went on for for a fairly long time, but at the end of the day, this didn't go to the market. Um, what do cyclodextrins compete with when it comes to this field? And these are the polysorbates, technically, who dominate the stabilization of the protein field. So there are a few, not a lot, but a few studies which compare the performance of polysorbates versus cyclodextrins. There is one particular cyclodextrin, the hydroxypropyl beta cyclodextrin, which is a bit in the front line of these studies, and that's partially because Rocket, one of the bigger cyclodextrin manufacturers, have uh, a product which is called Claptos Biopharma that is specifically suggested or proposed to be a component in the formulation of monoclonal antibodies. But it's not only this cyclodextrin, so there have been a lot of studies and a lot of papers published with other cyclodextrins. Of course, typically the ones that are approved for parenteral applications, that they can stabilize proteins, in some cases just as well as polysorbates. So Rocket is dominating the field. And they have shown using a lot of very relevant monoclonal antibodies and case studies that the proper formulation with a cyclodextrin can beat or compare or achieve comparable results to polysorbates. Why is it good to use cyclodextrins then? And what are the advantages, disadvantages? So polysorbates are not okay. They are used widely, but they are not ultimately safe molecules. So polysorbate degrade, they can form reactive oxygen species, which can cause a lot of nasty side effects within the body. So it's not an ideal excipient. And of course, cyclodextrins are very stable. They don't degrade, they don't metabolize, so they are exempt from such side effects or inconveniences. To also mention the drawbacks of the cyclodextrins, so polysorbates are typically used in the stabilizers in very low concentrations, sub 1% concentrations, whereas with cyclodextrins to achieve a stabilizing effect, you typically need or often need even up to 5 or 10% of the cyclodextrin. Um, so it could be even, I don't know, a 50-fold, 100-fold concentration difference between the polysorbate and the cyclodextrin. And also, in my experience, and I'm not saying that I've tried all the monoclonal antibodies existing, cyclodextrins do not always work. As we have learned, some of the effects the cyclodextrin have are based on a molecular interaction and need specific amino acids to be present and available on the surface of proteins. Of course, this is not happening all the time. So with some proteins, they do not work equally well whereas polysorbates are fairly general. An interesting aspect was then discovered, and it was also later patented that okay, there are the cyclodextrins, there are the polysorbates, and both of them can work to some extent. But what happens if we combine them? And if you are involved in the formulation of small molecules, you also know that some solubilizing agents can work very well with cyclodextrins, not as a competing species, but as synergistic species and molecules. So they have a combined synergistic effect. And this was also found in this case, as you can see, when they compare how the protein behaves upon stress, and you can see the control here and here upon steering, and then comparing the cyclodextrin and the polysorbate, you can see comparable effects, very different concentrations. But the interesting part is that when you combine the two, you get a much superior effect. So you add two types of mechanism of actions into the formulations, and what you achieve in the end is really fantastic. So this could be also a way forward considering, because of course pharmaceutical formulation do not have to compose of two or three components. You can easily combine for a better effect different components, and it seems to be working very well with cyclodextrins too. So apart from protein stabilization, how does effect on viscosity work? So this is an indirect effect that also comes down to prevention of aggregation. So the viscosity increasing effect of the protein formulations comes also from dominantly from aggregation and aggregate formation. And as we can prevent that with the cyclodextrins, we can also, uh, I wouldn't say reduce the viscosity, but not improve the viscosity. The good thing is that at this concentration that is applied, cyclodextrins do not contribute to the viscosity of aqueous solution. So it's the same as without the cyclodextrin. Um, Pre Preventing from surface absorption also happens similarly. So cyclodextrins can technically cover kind of the container wall, preventing the proteins from attaching to it. And also cyclodextrins, these cyclodextrins at least that are used here do not aggregate themselves, so they have a natural repelling effect from one another, so they wouldn't let the protein attached to the wall. As I said, unfortunately, I cannot really bring any commercial examples on protein formulations. The closest we have is glucagon, which is an oligopeptide. Uh, So it's of course a hormone, and it was fairly recently marketed as a nasal powder. So it's also one of the very few nasal powders that are cyclodextrin based and administered this way. In the parenteral field, the closest analogy is the tetrapeptide, so it's still a small molecule, but it's a blockbuster drug from from Amgen. Here, of course, in this case, solubilization is the dominant effect, so it's closer to a small molecule again. Okay, so that's about proteins. Let's see what cyclodextrins do with vaccines. Here, the examples will be much closer. Okay, they're actually on the market, so I can bring quite a few products that are either in clinics or already marketed with cyclodextrins. And as we'll see, surprisingly, how cyclodextrins are used in vaccines is completely different from their uses in protein formulations, because the first cyclodextrin approved was used as a vaccine adjuvant in a vaccine formulation, so not as a stabilizer or a solubilizer or anything else. This is a veterinary application, but conceptually it's the same as what would happen with humans. You can also recognize that this is a very peculiar cyclodextrin that we're talking about, sulfolipocyclodextrin, so maybe something that you have never heard of. And actually, if you're not familiar with this product, this is not surprising because this cyclodextrin is not sold by any company, not commercialized by any company. Actually, it's very difficult to learn anything um, from this particular cyclodextrin, even if you are looking very specifically. The product is rather old. It was approved in the 90s, I guess, so a long, long time ago. Um, and again, this sulfolipocyclodextrin was developed specifically to be the vaccine adjuvant. Of course, its structure is also closer a bit to vaccine adjuvant. So if you take a look at it, sorry, if you take a look at it, I hope you can see the mouse. Um, you can see that there are these strong aliphatic chains on the cyclodextrin, so it's a bit closer to an amphiphilic molecule or lipoic acid or something like that, not a typical uh, cyclodextrin. But the adjuvant effect was wonderful, and the product was super successful. So, not surprisingly, the second product that was on the market was also using cyclodextrins as adjuvants. What was surprising is that this was not anymore the sulfolipocyclodextrin that was used, but it was our old friend, common hydroxypropyl beta cyclodextrin. So this compound keeps surprising me all the time because, okay, first it was used as an excipient for oral formulations, then for parenteral formulations, and then it got used as a vaccine adjuvant. Okay, what kind of adjuvant effect could be associated with cyclodextrins? And then it's starting to be used as active ingredient. So what kind of molecule is this? Um, that can achieve all these features at the same time. So it always gets me thinking what can be the common points in all of these if there are at least dots to connect. Fascinating. But anyhow, hydroxypropyl beta cyclodextrin used as vaccine adjuvant in the product with a nice success. And of course, this opens again new doors because we have a commercial example where the cyclodextrin as adjuvant works, and this cyclodextrin is something that's commercially available. You can buy it under GMP fairly easily. So why don't we develop new products with hydroxypropyl beta cyclodextrin? And that's exactly what happens. And now we are crossing the line. So the products onwards are not veterinary anymore. These are human studies. So Daiichi Sankyo decided to explore a bit more how this particular cyclodextrin can be used as vaccine adjuvant. And what they developed was an intranasal influenza vaccine. And and luckily, of course, these are more recent studies. As you can see, it's very recent, just a few years ago. And accordingly, we have a lot of evidence trying to understand what is happening. if not on the molecular level, at least on the biochemical level. So what kind of immune response the cyclodextrin is triggering? And we have seen that it's enhancing lymphocyte proliferation. It keeps or it helps to maintain a longer immune response. It can also reduce the allergic risk of a reaction, but all the effects were correlated with T cell related immune response in general. And when considering the adjuvant effects of cyclodextrins, this is the closest that is uh, to the market. So we don't have any cyclodextrin-based vaccines where the cyclodextrin is used as an adjuvant on the market. This is in clinical phase 2, I think, this development. But apparently, since the effect is very general, I would assume that cyclodextrins as adjuvants would emerge. And this topic was really interesting for us, so we initiated a collaboration with Marseille University not that long time ago to explore. Okay, so now we have two cyclodextrins that we know to have adjuvant effects. So what is it that cyclodextrins aggregate, and what kind of cyclode activate in the immune cells, the monocytes, the lymphocytes, and what can we draw any structure activity relationship between the activation and certain biomarkers and the structure of the cyclodextrin. So with the help of this group, GP Gorwell, we screened a fairly huge library of cyclodextrins. As you can see, they were all very safe in these studies, and check different biomarkers in different type of cells. This is just a glimpse of the results, but as you can see, the differences between the positive control, and some cyclodextrins that we tested are dramatic. So in some cases, they do not trigger any immune response, and in some cases, the immune response that they trigger are bigger than the one of E. coli polyliposaccharide. I cannot really disclose the end of the story, but it certainly warrants the research of cyclodextrins as a novel class of vaccine adjuvants, which is one of my favorite potential new applications for these molecules. Okay, apart from vaccine adjuvants, what can cyclodextrin offers in vaccine development? So there was one product from Sanofi Pasteur where the cyclodextrin was used as a stabilizer, conceptually similar to proteins, we could say. So technically, it protected the inactivated virus. And it was it could also help in removing the use of preservatives. So they could develop a preservative-free and more stable product with the help of cyclodextrin, which is a nice success story. So this is again another application. And the most recent one is about using them as cryoprotectants. So, like many other sugar type molecules, cyclodextrins can also be used as cryoprotectants, and this effect has been recognized quite a while ago, typically associated with methyl beta cyclodextrin and cholesterol, um, how they could interact with the lipid rafts, and typically this application was exhibited for keeping different animal the fertility of different animal sperms for longer time during freeze and thaw process in banks and stuff. And it can also be used in humans similarly. But in this particular application, since cryoprotection is very critical for vaccines, and it has been extremely critical during the COVID times, it was not that surprising that one of the vaccines, namely the one from Johnson and Johnson, came up with HP beta cyclodextrin again as a component of the vaccine. And although not specifically said, from the information available, we can deduct that the cyclodextrin was contributing both to the cryoprotection and the stabilization um, effect of the vaccine. But what is also important to know that this application, the cryoprotection, can be also used in protecting other type of cells, not just sperm cells, during this process. So it can be used in CAR T cell therapies during the storage and the formulation of these compounds. So again, a new application that has a lot of potential. So, before we go into gene delivery, I would like to show some other biotech uses, and the reason for this bit strange setup is that these applications are still commercial. So these applications, although not mentioned often, they do exist, and some of the compounds that I'll show you are made with the help of cyclodextrins today. Whereas in the field of gene therapy, all the research is still preclinical stage. So this is more advanced. That's why I'm saying it first now. So how are cyclodextrins used in other biotech processes? These examples use cyclodextrins as additives, so not as components of the final product or components of the formulation, but like uh, boosters of the production, so that the yield, the outcome of the process is better and improved. And conceptually, there are a few different type of approaches. So in one approach, which we see on this slide, we often see that in an enzymatic process, one of the products, either a byproduct or the desired product, starts to block the process. So it acts as an inhibitor of the process itself. And as we said in the beginning, the cyclodextrin can mask the presence of certain compounds. Okay, the properties, sure, but even the presence of certain compounds. So if we apply cyclodextrins that can differentiate between the raw materials and the end product with a desired specificity, and they can encapsulate the end product, they can mask its presence, as then you can see uh, they can, upon in a concentration dependent manner, they can substantially improve the yield of certain production processes. Um, in another example, what we can also achieve with cyclodextrins is that we can have higher doses of the raw materials available for the process, which is of course a very straightforward application that can also contribute to an improved production. In some cases, cyclodextrins are also used for cultivation. Rarely, but cyclodextrins can also be used as feed for bacteria. But what's more important is that they can deliver some critical nutrients like vitamins, oils, fatty acids, whatever, to some sensitive bacteria, which can then facilitate the growth. And this is something that can be observed both in cell cultures as well as tissues. Um, the applications that's most fascinating is associated with the vaccine production, and this is how it's made today. And these are the pertussis toxin, the pertussis vaccines, both by Sanofi Pasteur and GSK. So in this case, a toxoid is used in the vaccine formulation, and the problem is that during the production, similarly to the first example, two inhibitors, two byproducts are generated, which can be encapsulated selectively by cyclodextrins. And here you can see how much the selectivity depends, or how important it is to have a selective cyclodextrin, because the performance of the different cyclodextrins differs substantially. And there is one particular cyclodextrin, again, not widely available globally, which is called a 2,6 dimethyl beta cyclodextrin that could boost the toxoid production 100-fold. And this here we are not talking about a huge amount of the cyclodextrin, just 1% added to the reaction can boost the production process 100-fold. And this is an amazing feature. This particular cyclodextrin is a methylated cyclodextrin, as I said, but it has a unique structure. As you can see, the methylation pattern is not random, like with the common excipient cyclodextrin, but it's very specific on the 2 and the 6 positions in the compound. So this molecule is not easy to make, and of course, its performance in this particular production process is very different from the common random methyl beta cyclodextrin. So, a few words about gene delivery, which is an upcoming but very promising technology with cyclodextrins. So, uh, okay, what kind of options we have when we want to deliver genes? Now, of course, everyone is an expert in gene delivery since 2020. But of course, we have two basic technologies. We have the viral vectors, which is the older technology, and that's also how some vaccines were developed. As far as I remember, the AstraZeneca and the Russian vaccine, maybe also the Chinese one, contain the viral vectors, and there are non-viral vectors of different types. And both have advantages and disadvantages, but currently the regulators are leaning towards non-viral vectors and their use in the new type of formulations. So how cyclodextrins can be used in this kind of formulation? Cyclodextrins are extremely versatile and fairly easy to combine with other drug delivery systems. When we talked about small molecules, we discussed it a lot, but technically they can be incorporated into to micelles, liposomes, nanosponges. You can form polymers. You can combine them with different polymers. And these are more or less the technologies that are used to deliver genes. Uh, so you can add cyclodextrins into such formulations, and that was the original strategy to improve not necessarily the performance, but certain characteristics of these formulations, the solubility, the safety, the tolerability, and so on. So, uh, use cyclodextrins in small amounts to improve the drug delivery system, but it was not the critical part of the drug delivery system, not the part that was interacting with the oligonucleotide. That happened later. So what I talked about before is this part where they are enhancers of the formulation. There are other technologies where the cyclodextrin itself can carry the oligonucleotide, and we distinguish four different strategies. So, cationic cyclodextrin polymers, similar to the concept of polyimine, polyrotexanes, which are these necklace-like cyclodextrins, um, pure cyclodextrin vectors. You can see that these are amphiphilic cyclodextrins with a long aliphatic chain on one end or hydrophobic chain on one end and the cationic groups on the other end. These compounds can, of course, formulate, not of course, but they can form micelle-like structures, particles that can encapsulate within their cavity the target molecule via these ionic interactions, of course. And there are these supramolecular structures of cyclodextrins, which conceptually work similar to polyrotexanes, but this is not a polymer-like structure, but individual cyclodextrins interacting with one another to form this assembly that can capture the DNA. What we work on at CarboHyde and where the most of the literature is are these two areas, the polymers and the pure vectors. The good thing with the polymer is that the chemistry is very nice and very versatile. You can add all kinds of apelide groups, you can add a lot of cationic groups, you can modify the density of these groups within the structure, even combine different types of substituents. So you can come up with a wide versatility of polymers. And these polymers were evidently shown very nice uh, to interact with the oligonucleotides and deliver them into cells. We also have a few nice papers with the University of Debrecen on this. So these are called polyplexes, and they have a lot of advantages. Of course, this there are also some drawbacks, mostly related to the uptake. So how excessive of a knockdown you can achieve with the system. What is the cargo that you can deliver into cells with the polymers we have observed, or everyone observed that these are limitations, and also there were some issues with toxicity and immunogenicity. The other possible application is to use amphiphilic cyclodextrins, and of course here the chemical modifications are also very vast, and there can be a lot of compounds created, and as I said, these form nanoparticles that incorporate in their uh, inside different types of oligonucleotides. Again, we have dealt with this research quite a lot, and we explored how the differences in the alkyl chain and differences in the ionic groups affect the delivery. Since publications are on the way, I cannot really disclose how far we got with this. What I can say is that we worked on this, so this is a European grant here, an EU Horizon grant, aimed to deliver and to create new drug delivery systems for gene therapy. The peculiarity is that it's for oral therapy, so it would be one of the first oral therapies. And we created a huge library of cyclodextrins, so now we have a lot of knowledge of what works and what's not working, and also what kinds of gas molecules we can incorporate. But clearly, what we have seen, and this is not from this study, but a former one on Huntington models. What we have seen is that, uh, of course, this is antisense oligonucleotide. So the oligonucleotide in itself cannot really enter the cells, but when it's incorporated into the cavity of these cyclodextrin-based nanoparticles, it can get into the cells. And as you can see, the different types of cyclodextrins have um, different efficacy, and with some of them in this case we could reduce the level of the target protein by half, so to about 50%. Now since then, in the GeneGuide project we could achieve over 80%, so 80 to 90% knockdown, which is comparable to the efficacy of lipid nanoparticles. So in general, we consider this application, the amphiphilic cyclodextrins, much better. Now we have a lot of tremendous preclinical data showing that they are very safe, and they have very limited toxicity and immunogenicity issues. Uh, Also, these compounds are fairly economic to make, and very robust procedures can be established. So we already did some scale up and getting closer to animal studies. And one other additional advantage of the compounds over the currently used systems is the size-wise limitation. So originally we were working with smaller oligonucleotides, typically plasmid DNA, the antisense that I showed, and siRNA. But now we are exploring bigger oligonucleotides like mRNA, and that's one of the first occasions that cyclodextrins are uh, are explored if this can be done, and the results we have are very positive. So I think that there could also be a nice future application for a novel class of cyclodextrins. So as you can see, unlike with the former examples, here we are talking about necessarily new chemical entities, new excipients. The cyclodextrins that are used in protein and vaccine formulations currently are not capable of delivering oligonucleotides. So this is the reason why this research is much behind compared to other formulations with the biologics. And this is what I wanted to say. So these are the four main topics, and I think each of them are very fascinating. And at CarboHyde, we are particularly interested in all of them. And if you are into working with biologic formulations, I hope that the discussion and the examples today could convince you to at least put cyclodextrins into the toolbox that you have for biologic formulations because, well, they may be able to surprise you. And with that, I'll conclude my talk. So, thank you for listening and the attention. So, I stop sharing. And okay, so we have some time. So, if you have any questions related to the topic or again, unrelated to the topic, just type into the chat box and I'll try to answer. Okay. So, I don't know, is but Henry asked, is it visible to anyone? I think it is, but it's about the business strategy of CarboHyde. So I can provide only a limited number of information. We don't really have a catalog of these products. The cyclodextrins for gene delivery are more or less custom made. We have a few candidates that we identified already that we know that work very well, but these are not available commercially. So if someone is interested in evaluating those for research, you can drop us a line. We have collaborations in that field. And I forgot to add that what we also found, and this is not our founding, but okay, so these are amphiphilic cyclodextrins capable of creating nanoparticles. But the nice part is that in these nanoparticles, you cannot only incorporate large molecules like biologics. We have collaborations, and there are groups that have shown that small molecules can be incorporated into these cavities and use them for cancer therapy, or even more so, the combination of small molecules and oligonucleotides could be incorporated into the cavity of these small molecules. So with amphiphilic cyclodextrins, I really think that the possibilities are endless. Apart from that, for vaccines and biologics, typically these are the commercially available cyclodextrins that are usually used. Major competitors in the market. So these are upcoming applications of cyclodextrins, and accordingly, I would say that in all cases, the main competitors are the mainstream solutions. So for protein stabilization, the main competitor of cyclodextrins is polysorbates. For vaccine adjuvant, it would be the aluminum salts and the currently used typical vaccine adjuvants. And in every case, cyclodextrins could be listed, of course, a lot of benefits. There are, of course, also some drawbacks. You need fairly high doses of the cyclodextrins. They are big molecules. They are not necessarily the cheapest. But on the other hand, using cyclodextrins in such formulations very often offers new IP. So you can have IP protection. It's not as trivial to use cyclodextrin in a as a vaccine adjuvant or in a protein formulation as it would be to polysorbate. So gaining intellectual properties is usually an added value when working in cyclodextrins in this field. And in the field of gene therapy, the competition is, of course, lipid nanoparticles. Uh, have your products been tested in larger biological contexts like organoids, spheroids, 3D bioprinting applications, or tissue regeneration? So, uh, I'm not exactly sure which of the applications we are referring to. Obviously, the proteins and vaccines are much beyond that, as there are commercial products. In vivo data is available. For the gene therapy, I think in our group, for sure, only cell culture data is available. But from former studies, there are also in vivo animal data that has been generated, and it also underlines the potential use of cyclodextrins in gene therapy. So yeah, there are data in also more complex systems, not always with our candidates, but conceptually it's available. Any further questions, maybe? I'm just scrolling if I missed any. No, but I don't think so. Um, okay, if you don't have further questions, then I think we can uh, stop the Zoom call. But in any case, if you have, okay, maybe is there a need for smaller or larger CDs? Well, then the one which are available, so smaller cyclodextrins than the alpha would compose of five sugars, and although there has been a paper that it's possible to make, technically it's not economic, and even the alpha cyclodextrin's cavity is very small, so only a aliphatic chain can fit the alpha cyclodextrin cavity. A cavity smaller than that, I'm not sure what it would fit into. That would be still hydrophobic because like ions cannot really fit well into cyclodextrins. Larger cyclodextrins than the gamma do exist. So cyclodextrins composing of 9, 10, even up to 30 sugars. But currently, so these are not natural molecules, at least in this in reasonable quantities. They are not produced by the same enzymes as the alpha, beta, and the gamma cyclodextrins. So usually you have to make a very specific process to enrich these products in the enzymatic process, usually some templates, or you have to tailor the enzyme to create these cyclodextrins. But so far, these efforts were very limited and they couldn't really generate reasonable amounts of the material. So there is just simply no data for the application. We there is not a need because we don't know what these cyclodextrins could be used for, but probably something different than the current molecule because they don't really possess anymore this ring-like structure. The the cavity is not as solid as with the current cyclodextrins. It's like a bit fluent, waving, and stuff like that. So I'm not really sure if that's more like research fantasy or there is an actual use for those compounds. At least we don't know that today. Okay, if you have any questions in your mind, just feel free to drop us a line. Yeah. Okay. Okay. So, do you support the use of dilated cyclodextrins to deliver treatments to the brain via the nasal pathway? So, by support, do you mean if we make dilated cyclodextrins or think it's a good idea? Dilated cyclodextrins are fairly peculiar because they have these mucoadhesive properties. So, in a lot of applications, mucoadhesive uh, effect, or this is certainly an additional value to cyclodextrins that they can offer. So intranasal applications, yeah, certainly, I would say if a cyclodextrin can stick to the surface of the application in such case and thereby deliver uh, their locally a higher concentration of the active, For sure, it's something to be considered seriously, or for an eye drop. Okay, it can be similarly very useful to develop. The problem with the dilated cyclodextrins, so okay, that's also a new class of excipients. So the road is very long for such. But I always say when people ask me, okay, so will there be new cyclodextrins as excipients? And what I always say is that no new cyclodextrins will be, in my opinion, marketed that can do exactly the same as the ones that are available now. So there are seven cyclodextrins which are on the market, and all the seven has exactly the same mechanism of action. They can work on different molecules, but what they do is exactly the same. Now, then I add that if we consider cyclodextrins that can do more than that or something different, those I see as reasonable potential new excipients, like the amphiphilic cyclodextrins to deliver gene or small molecules forming nanoparticles, or something that can be used as targeted delivery, or like like you proposed, Charbel, thiolated cyclodextrins that have mucoadhesive properties. That's something good, something that has a long road with all the safety studies and optimization of the structure. But yes, I think it's a reasonable research to pursue, and something that's also in very good hands in Innsbruck. Okay, so I'm happy that everyone liked it. Our time is up, but again, if you have any further questions or want to discuss further, I'm really happy to answer any questions. And you will receive the recording of the presentation probably tomorrow, or at least very soon. And feel free to share it with your colleagues. We'll put it on YouTube and whatever, so it's free for use. It's educational content. We are happy if more and more people see it. So thank you for your time. It was nice having you. Enjoy your evening, your day, wherever you are. So have a good one. Bye bye.